We are studying biochemistry. We learn DNA damage and repair. My name is Anna Ogaito.
I'm Ben Schroeder.
I'm Sophie Rhodes.
I'm Yukio Mimono.
To fully understand the concept of DNA, we first looked at the structure of it. So in the top right corner, it's just a simple nucleotide. So the green circle is the phosphate, the blue pentagon is the deoxyribose sugar, and then we've got the base. So there's 4 different types of bases. We've got the thymine, the guanine, cytosine, and the adenine. So there's 3 hydrogen bonds between the cytosine and guanine, and 2 hydrogen bonds which form between the adenine and thymine. And there are 10 bases per turn of helix, and diameter is 20 Armstrong.
Radiation is a process in which the energetic particles or waves travel through a space or medium. It is spelled into 2 general groups, ionizing and non-ionizing.
放射線とはエネルギーをたくさん持った粒子が媒質や空間中を伝わることです。放射線は大きく2つのグループに分けられます。原子や分子を電離させるものとさせられないものです。
There are 2 examples of ionizing radiation, which are X-rays and gamma radiation, and these cause damage to biological molecules.
So the other group is non-ionizing, and this effectively means that you, funnily enough, won't ionize. The examples of these include UV, infrared, and microwave radiation. So how does this cause DNA damage? They're commonly evident in terms of mutations. So within the DNA code, you have little triplets which make up the coding. If these mutate, then the protein it codes for will take on different properties, and this can lead to cancer.
Muticals は、変容という意味を表して、変わりがない、This one is、発がん性という意味です。
Okay, so we didn't really know the answer to this question when we were asked it, but do you think DNA damage is common in the body? Anyone? Okay, we'll answer it. It's actually quite common, but not to worry, because we have the DNA damage response, which is your body is pretty good at normally.
Franz Sanger determined the first sequence of insulin in 1952. The biochemistry department, in which we received interesting and engaging lectures and discussions by Dr. Takashi Ochi, Dr. Kwon Woo, and Professor Tom Blondell.
As well as Sanger, Watson, and Crick also researched at Cambridge. They discovered the double helix structure of DNA. A lot of their works in the EGOPUB, probably alongside a couple of pints.
So this is one of the mechanisms in which your body will repair DNA. So you've got the X-ray radiation coming in, which is not good. But so then you get the double strand break. So now you've got the break, but because they're complementary, what they'll try and do is find a site on the other one, which will fill in the gap, which is what they're doing here. Then other enzymes turn up, DNA polymerase. There'll be some free nucleotides just hanging around. They'll be able to put them in there, and then we can get our lovely DNA back intact. Okay, so now that we've found the interesting proteins and we know what they do, but we don't know how they do it. So one way to do it is to try and find out their structure. And to do that, we have to go through these 3 general steps. So cloning, we have to try and take part of the gene we want. And then we have to try and go through quite a complicated process where we end up having to put it within inside a plasmid. And then we can eventually get the gene we want. And this involves what's called PCR, which we're about to talk about in a minute. You then have to purify it because other proteins will still be made and you don't want them. And then characterization is where you look at it and you figure out what it's doing, and yeah.
So when we want to do PCR, we have to find out about the proteins, and then we purify and then crystallize. And PCR stands for polymerase chain reaction. So the heat's just broke up the strand, but so the hydrogen bonds have broken. So then this is cooled, and is it like this?
Okay, so the primers turn up on the genes we want. The polymerase then comes along and starts fixing the other nucleotides to the rest of the DNA. So it has to be cool for this part. We then heat it again, so to break the hydrogen bonds. And then we just keep repeating the cycle because we want to amplify the gene, so we just want loads of it, basically. So we keep doing this and doing this and doing this. So get rid of it.
So we were given a case study to put our newfound knowledge into practice. So where it says gel filtrations, they like separate proteins according to the size. They separate like the bigger proteins and then the smaller ones are last. The MAU, which is along the y-axis, is the UV absorbance. And the different colours represent the different proteins. So as you can see, the green one, that's that XLF XRCC4 1-to-1, was a mixture of proteins. So that's bigger, so hence the like peak shift to the left. So the bigger molecules come out faster, so can go through the larger pores. So the smaller molecules, meanwhile, can go through all the pores, so they take longer, which is just what the picture shows. So here is an image of an example of things we saw through the microscope, which is in the top left. So you can't see a protein using light, as light has a wavelength that is too long, but we can use X-rays because they have a shorter wavelength. So to be able to see the proteins, we need to make a crystal in which the proteins are homogeneous. So the image is not distorted by the other proteins and not too flexible. We can see the actual protein structure using a process called NMR, which stands for nuclear magnetic resonance.
So you'll notice the weird physics picture on the lower right, and we use this because the intensity of scattered light from a single point, which stands for a single protein, is just not enough to be able to really determine protein structure. So we use something called Bragg's law, which is what we're shown here, to our advantage, where we essentially measure the intensity of the scattered light that satisfies this law, and we produce quite a few dots, as you're about to see.
The X-ray generator we saw at the labs had a wavelength of 1.418 angstrom. As X-rays hit the crystal, we needed something to keep it cool. This is the cryostream. This keeps the temperature constant and low, which prevents damage to the protein crystal.
私たちが実験室で見た X 線の発生器は 1.5418 オームストロングの波長を発生させます。X 線が結晶を熱するので冷やしておく必要があります。このための装置をクライオストリームと言います。これは温度を一定に低く保ち、タンパク質の結晶へのダメージを防ぎます。
So this is basically what we've just gone through. So you have your crystals, and then by X-rays you get your diffraction pattern, and then they use a software on the computer called electron density mapping, which is useful for biochemists as they can like see all the peptide bonds and see the structure.
Okay, so I'll talk you briefly through protein structure, where the primary structure is where you have the amino acids in a row, joined by peptide bonds. The secondary structure is essentially, because of the primary structure, you'll create various intermolecular forces, which will make it either form an alpha helix, beta pleated sheet, or omega loops. This then determines the tertiary structure, which is the first one to be in 3D, and is basically an exaggerated secondary structure as it progresses. The intermolecular forces make it further coil and whatnot. Quaternary structure may sound scary, but it's essentially several polypeptide chains in tertiary structure. So that's all it is, really.
In conclusion, proteins have various functions within the body: enzymes for digestance, antibodies for fighting infection, and the structure compounds.
So what have we learned from this? We learned how important protein structure is in medicine. So when you're trying to make a drug, it obviously helps if you're figuring out what you're trying to do. We further improved our communication skills. and we've catalyzed our interest in biochemistry. Slightly cheesy, but yeah.
We would like to take this opportunity to thank Barclays and Rolls-Royce for funding this amazing week that we are all lucky to have been on. Also, thank you very much for Dr. Eric Albone for the organisation of this week. We have all thoroughly enjoyed it, and we have taken some amazing memories from it too. Thank you to all our teachers and everyone involved in making it so memorable.